A very warm greetings to everyone. Today I, Khyati Sharma,
And I, Tanisha Joshi, have prepared a project on the topic Food You Eat, Adulterated or Not. The sub theme of our project is food security.
under the guidance of Mr. Rahul Vadera sir.
Our project's topic is Food You Eat. Is it adulterated or not?
But before, let's look through the content of our project.
Food adulteration.
Recent advances in detection of food.
Food poisoning and intoxication, a global leading concern for human health.
Chemometric methods for spectroscopy based pharmaceutical analysis.
The Indian scenario.
Adulteration test.
Detection of water in milk.
Detection of artificial color in turmeric powder.
Detection test of butter or ghee.
Detection of original honey.
Detection of white powder in ionized salt.
Let's first of all look what is food adulteration. Food adulteration can be defined as the practice of adulterating food or contamination of food material by adding a few substances, which are collectively called adulterants. Adulterants are the substance of poor quality products added to food items for economic and technical benefits. Addition of these adulterants reduces the value of nutrients in food and also contaminates the food, which is not fit for consumption. These adulterants can be available in all food products which we consume daily, including the dairy products, cereals, pulses, grains, meat, vegetables, fruits, oils, beverages, etc.
Moving further, now let's talk about recent advances in the detection of food. Food adulteration is a curse for mankind. It has been happening since times immemorial and continuing in the recent times. To control such problems, various technologies have been developed. In the present work, the detection methods of food adulteration are reviewed with a focus on detection of common food adulterants.
Since food adulteration is a huge concern in all parts of the globe, there is voluminous literature on various aspects of food adulteration, including its detection. In fact, methodologically, procedures of food adulteration detection have been reviewed extensively. Many of the methods for detection of food adulteration require elaborate steps of sample preparation, prior analysis involving high-end technologies that makes the whole process difficult to perform and time consuming.
Therefore, considerable interest has emerged in developing rapid methods for food adulteration detection. Thus, rapid online detection of food quality is a non destructive manner because even more relevant.
Food poisoning and intoxication, a global leading concern for human health. Food, the major fundamental unit of living life, has several types of intrinsic and extrinsic factors such as hydrogen ion concentration, water activity, redox potential, different types of nutrients, antimicrobial substances, temperature, and environmental conditions for storage that regulates the growth of pathogenic microorganisms responsible for food spoilage, poisoning, and toxicities, as well as foodborne illness.
Sometimes food may also become poisonous due to the ingestion of some toxic chemicals or may be contaminated by pathogenic microbes due to the mistreatment of food, improper way of food processing and preparation, mixing of different types of flavoring agents without knowing its ingredients and reactions, intake of reheating food, unhealthy cooking and consumption policy, consumption of already contaminated food or foods infected by insects, use of the excessive amount of preservatives or improper way of storage which may be the main causes of sickness.
Chemometric methods for spectroscopy based pharmaceutical analysis. Spectroscopy is widely used to categorize pharmaceutical products or processes, especially due to its desirable characteristics of being rapid, cheap, non invasive, non destructive, and applicable both offline and in at online. Spectroscopic techniques produce profiles containing a high amount of information which can profitably be exploited through the use of multi-rate mathematic and statistic techniques.
The Indian scenario.
The Food Safety and Standards Authority of India recently proposed to set maximum levels for lead for more food categories in addition to those mentioned in the existing regulations. While these limits are aligned with the initial limit set by the Codex, FSSAI will now be expected to focus on the revised lead standards too.
Indian regulations on antibiotic use in food producing animals are largely ineffective. In July 2014, Delhi based non-profit Center for Science and Environment conducted a study that revealed indiscriminate use of antibiotics in the poultry industry and non-therapeutic purposes. Such indiscriminate use of contributes to the development of antibiotic resistance, a major global health concern today.
Adulteration test. First comes is detection of water in milk. First put a drop of milk on a polished slanting surface. Pure milk either stays or flows, slowly leaving a white trail behind. Milk adulterated with water will flow immediately without leaving a mark.
The second adulteration test is detection of artificial color in turmeric powder. Add a teaspoon of turmeric powder in a glass of water. Natural turmeric powder leaves light yellow color while settling down. Adulterated turmeric powder will leave a strong yellow color in water while settling down. Here we are mixing the pure turmeric powder in the glass of a water, and secondly we are mixing the adulterated turmeric powder. The pure turmeric powder leaves a slight yellow color in the glass of water, whereas the adulterated turmeric powder leaves a whole yellow color.
Detection test of butter ghee. Take a teaspoon of melted ghee or butter in the test tube or transparent bottle. Add a pinch of sugar, close the container and give a vigorous shake. Let it stand for 5 minutes. If a red color appears at the bottom of vessel, then the sample contains vegetable oil.
Here we are mixing 1 teaspoon of sugar in the pure ghee. Secondly, we are mixing same a teaspoon of sugar in the adulterated ghee. We can see after leaving the glass for 5 minutes that adulterated ghee leaves red particles lying at the bottom of the glass. Detection of original honey. Add a spoonful of honey to a glass of water. If the honey disappears instantly, it indicates the presence of glucose or sugar syrup. Pure honey is denser and will instead sink to the bottom instead of dissolving instantaneously. Another test involves mixing a tablespoon of honey, a little water, and about 2 to 3 drops of vinegar together. If the mixture results in foaming, it indicates possible adulteration in the honey.
Here we are adding 1 teaspoon of pure honey and again we are adding 1 teaspoon of adulterated honey in a glass of water.
Pure honey settles at the bottom of the glass, whereas the adulterated honey leaves yellowish color in the glass of water.
Detection of white powder in iodized salt. Stir 1 by 4 teaspoon of sample of salt in a glass of water. Pure salt dissolves completely and gives a clear solution or gives slightly turbid solution due to the presence of permitted anti-aking agent in the salt. If salt is adulterated, solution turns dense, white, turbid in the presence of chalk powder and other insoluble impurities will settle down at the bottom. Here we are adding 1 teaspoon of pure salt and again adding another teaspoon of adulterated salt in glass of water. The pure salt gives us a crystal clear solution, whereas the adulterated salt gives a white colored solution with settlements at the base.
A whale of food testing. What would happen if we didn't have regulated food testing in place?
Though it may be the last process of food manufacturing chain, food products testing is the most important step because it determines the safety of the food for use.
Food product testing is vitally necessary to ensure that the food is free of physical, chemical, and biological hazards. Examples of potential hazards, food contaminants include metals, E. coli bacteria, salmonella, cleaning agents, additives, preservatives, pesticides, and more. So, here we both end with our project work. Thank
Thank
you.
you.
Stay healthy and eat healthy.